Good afternoon, everyone, and welcome to this week's National Bleeding Disorders Foundation Wednesday webinar. My name is Fiona Robinson, and I am the series host. The title of today's webinar is HLA alleles associated with inhibitor development in severe hemophilia A. This webinar is being recorded. The recording will be available shortly after it ends on the NBDF website, bleeding.org. We encourage you to ask any questions you may have today. You can ask your question by using the Q&A feature at the bottom of your screen at any time, and we'll keep an eye out for your questions, and then when our speakers have completed their presentations, we will put your questions to them. Today, we are very fortunate to be joined by a whole panel of medical and lived experience experts. Dr. Michael Recht is the Chief Medical Scientific Officer at the National Bleeding Disorders Foundation, and an investigator on the My Life, Our Future study, and a faculty member at the Yale Center for Bleeding and Clotting Disorders. Dr. Kathleen Pratt is Professor and Vice Chair for Research in the Department of Medicine at the Uniformed Services University of the Health Sciences in Bethesda. Dr. Sarah Contante is an Associate Professor in the Department of Pathology, and Dr. Anwar Ahmed is an Associate Professor in the Department of Preventive Medicine and Biostatistics, and they are also both at the Uniformed Services University of the Health Sciences. Dr. Mosi Williams is a lived experience expert, a person living with severe hemophilia A and an inhibitor, and a social worker at the University of California, San Francisco Adult Hemophilia Treatment Center, and also a research participant. I'd like to thank all of our expert panelists for joining us today, and I'll pass things over to Dr. Recht now to get us started.
Thank you very much, Fiona, and welcome everybody to today's webinar. Today we're going to be talking about My Life, Our Future, the Factor VIII genotyping program that was and is a nationwide United States-based initiative that was launched in 2012 through a partnership between what was then the National Hemophilia Foundation, now the National Bleeding Disorders Foundation, BloodWorks Northwest, and the American Thrombosis and Hemostasis Network. The initial funding came from Biogen. The goal of My Life, Our Future was to offer free genetic testing to people with hemophilia A and potential impacted family members, with the goal of identifying specific Factor VIII gene variations that could cause hemophilia. The program not only provided participants with valuable information for clinical care and family counseling, but also created a large de-identified research repository to advance the understanding of hemophilia genetics and treatment, response to various treatment, and the risk for developing an inhibitor. Today, we're going to hear the work from Dr. Pratt and Dr. Cantante and Dr. Ahmed using the My Life, Our Future research repository. But before we start that, I'd like to talk to Dr. Williams about just his thought as a lived experience expert, his thought about the importance of research, participating in research, and participating in the My Life, Our Future program.
Thank you, Dr. Michael. My name is Mosi Williams. I am a person with severe hemophilia A with an inhibitor that is now tolerized. And so my experience as a 47-year-old and having lived through the bad blood epidemic and seeing now how I, having a tolerized inhibitor, have benefited from participating in research and studies and clinical trials and seeing the changes in the landscape in the hemophilia community in terms of treatment, particularly for individuals with inhibitors. As previously there wasn't. I can remember not having factoring, my mom telling me not to take what was available because of the concerns about the blood in the system. And so now being a provider, a mental health provider, a social worker at a hemophilia treatment center, I'm able to share my experience and encourage others, inform them about research and participating in how it can benefit. It has benefited us as a community currently and can also pass on to the next generation. As a Black man with hemophilia, I think of my own ethnic background and some of the history of research that was misused against the Black community. So there's hesitance from some people to participate in research for that reason. And so I try to use my experience and also looking at correct information and making sure people have an informed, are completely informed about the processes now, being safe, so that they aren't mistreated, so that they understand completely what they will be participating in and how it can benefit them, potential risk and all those things. And as I share that, and then also talking with the rest of our team, our medical provider, to talk with them as well and to kind of dispel some of the stereotypes about current research and efforts.
Thanks so much, Mosi. I really appreciate your input. So now I'd like to pass it on to Kate and Sarah and Anwar to learn about how they use the My Life, Our Future research repository and what they found. So thank you. Take it away.
Okay, thank you very much. So it's just a real privilege to be invited to present a webinar for the National Bleeding Disorder Foundation series. And it's been a privilege for a while to have access to the My Life, Our Future database and the associated Athens database, et cetera. So thank you all very much. And it's always just so inspirational, especially to hear from people with lived experience who will give us a thumbs up. So thank you. Thank you all. So again, the title is HLA alleles associated with inhibitor development in severe hemophilia A, analysis of the My Life, Our Future hemophilia A cohort. This is our standard government disclaimer. The three of us presenting today are all government scientists. Nothing you hear is an official communication from the US government or from our respective institutions. Regarding conflicts of interest, I've over the years received honoraria, travel reimbursements, and some grant funding from several companies that produce factor VIII. I'm also an inventor on several patents related to factor VIII immunogenicity, but none of these are directly related to this study. The other authors have no conflicts to report. So what are we going to talk about today? We are going to talk about why we are doing this research. We'll go into a little bit how we did it. We'll show you some of the results, what we found, and finally at the end, a few thoughts of what it could mean for the hemophilia A community. So here is a complicated slide. I'm not going to go through every detail of blood coagulation here, but I want to point out how important factor VIII is in blood coagulation. So there are two arms to the blood coagulation cascade, meaning all the enzymatic reactions that happen in your blood to make it clot. You have the extrinsic pathway, which kicks off when you have a wound and you have bleeding, and these factors in your blood come together to try to stop the bleeding. And when the extrinsic pathway kicks off, it eventually generates a small amount of thrombin. Some of that thrombin will then activate additional enzymes in your blood and start the intrinsic pathway over here. So again, the extrinsic pathway initiates coagulation. The intrinsic pathway accelerates it. Well, how much does it accelerate it? Well, first of all, why does it accelerate? There are a number of acceleration phases, but the most important one involves factor VIII. So there's an interaction right here that links intrinsic and extrinsic and increases the clotting process by 4 orders of magnitude. In other words, blood that has factor VIII will clot 10,000 times faster than blood without factor VIII. That tells you right there why severe hemophilia A is a serious bleeding disorder. So why are we doing this particular project? Well, from a high level view, the standard traditional treatment for many decades for those with severe hemophilia A has been infusions with factor VIII several times per week. Recently, there's been, as probably almost all of this audience knows, a real revolution in the different therapies that are available for hemophilia A, including a bispecific antibody referred to as emicizumab or Hemlibra. And this is a wonderful therapeutic. It has to be infused much less frequently than factor VIII. But hence, the overall exposure to factor VIII may be delayed for quite a while and is often quite limited compared to prophylactic factor VIII therapy. All that said, if you have a serious bleed or an upcoming surgery or dental procedure, factor VIII is still very strongly preferred to treat bleeding in those kind of scenarios. Therefore, even if you're someone who has chosen the bispecific antibody or who might choose some of the other non-factor therapies that are coming on board soon, the development of neutralizing anti-factor VIII antibodies, referred to commonly as inhibitors, is still a major concern. For decades, inhibitors have been treated by immune tolerance induction, which is just a fancy way of saying intensive factor VIII therapy until they hopefully subside. This is a very extraordinarily expensive process, has a success rate of about 70%. So we still need to better understand this immune response to factor VIII, why it occurs in some people, not others, so that we can come up with better ideas on how to avoid or prevent it. So this talk focuses on human leukocyte antigens, abbreviated HLA. What are HLA? These are specialized proteins that are found on the surface of many of our cells, and they basically can distinguish between self and non-self. In other words, between the molecules that make up our body and invaders that we don't want there. HLA proteins are essential for the development of antibodies. They're encoded by HLA genes on chromosome 6 in humans, and this is by far the most variable region of our entire DNA. Indeed, there are thousands of potential different HLA variants or genes. We'll use the terms genes and alleles interchangeably in the talk. They both are in common use. So there are many, many possible variants, but each of us carries about, depending on how you define HLA, about 13 to 15 HLA genes, and we have 2 copies of each. Why do we have 2 copies of each? We get one from mom and one from dad, basic biology. So this is a cartoon I like to show of the initiation of the immune response to factor VIII. You have antigen-presenting cells in your body. If they encounter a circulating factor VIII, that's a picture of the factor VIII structure there, factor VIII will be taken up by the cell, chopped into its component pieces. These are referred to as peptides. If one or more of these peptides then binds to an HLA molecule, it can be transported to the surface of the cell, where it can encounter a circulating T cell. T cells have T cell receptors, abbreviated TCR here, that recognize the HLA bound to the peptide. When this very specific recognition process happens, you get stimulation of the T cell, which then secretes molecules called cytokines. Those lead to development of plasma cells that secrete antibodies. So this is now zooming in on a small part of what I showed you on the previous cartoon. This is the HLA again. This is the T cell receptor again. And this is the piece of factor VIII, or whatever the foreign protein is. And the point of this picture, which is from a very nice immunology textbook, is to show you that there's a specific shape to a groove on the surface of HLA. And there's also a specific shape to a groove on the surface of the T cell receptor. And these specific shapes can only accommodate particular shapes of the peptide from factor VIII or whatever your invader is. And these need to have the proper shape and the proper electric charge in order to make a high affinity complex here, leading to T cell activation. These peptides that bind the HLA and stimulate the T cells are sometimes referred to as epitopes. You might hear that term later. So how does HLA contribute to inhibitor risk? Well, factor VIII is a foreign protein if you're an individual with severe hemophilia A. Interestingly, even patients with a single amino acid difference between their circulating non-functional factor VIII or dysfunctional factor VIII and therapeutic factor VIII, these individuals may or may not develop an immune response to infused factor VIII. The reason that those who develop an immune response do develop that response is because of exactly what I showed you on the previous slide. There was one peptide, one region in the factor VIII, the therapeutic factor VIII, that was different from their own factor VIII, and it bound to an HLA and stimulated the T cell. So CD4 T cells are the variant of T cell that's needed for an antibody response, for initiation, for stimulation of the B cells, for maturation of the antibody response. So in our study, we are studying demographic, clinical, and genetic data from individuals with severe hemophilia A who participated in the My Life, Our Future research repository. They agreed to have their genomes sequenced, and they also shared their clinical demographic phenotypic data with the Athens dataset. So in other words, if you participated in both Athens and My Life, Our Future, your data was used in this study. We needed data from both ends there. We used the whole genome sequence data then, as Dr. Contente will describe in a bit, to determine the specific HLA alleles that each individual carried. We next asked if any of these alleles or combinations of HLA alleles correlated with inhibitor development. Dr. Ahmed will go into that for a bit. I want to point out that the entire statistical analysis was only possible because of the large number of volunteers who agreed to participate in the My Life, Our Future project. If you're going to do statistical analysis and you're asking a pretty complex scientific question, you need a large amount of data and a large number of individuals contributing their data and/or samples. So again, thank you to everybody who contributed. I'm waiting for the slide to advance. Okay, Dr. Contente will take it from here. You're muted, Sarah.
Thank you. Thank you, Dr. Pratt. Good afternoon, everyone. The DNA sequences for each individual are stored in a secure repository of the National Heart, Lung, and Blood Institute as part of the Top Med program. We received permission to download and use this information. So each of the 1,841 genome sequences were downloaded and then analyzed using a software called HLA. This software program takes the region on chromosome 6 where the HLA genes are located and compares the sequence with a sophisticated graph structure that represents all of the thousands of known HLA gene sequence variations. Based on this comparison, it reports out the name designation of each allele found. The designation includes the letter name of the gene, A, B, C, and so on. This is followed by 2 digits that specify the allele group, then 2 digits to indicate any protein variation, and then 2 digits to indicate any sequence changes that do not affect function. So this is the 6-digit resolution. We'll see some examples in a moment. The statistical tests on the HLA data were performed using software from SAS. We're trying to understand why some people develop inhibitor after factor VIII treatment and others do not. We compare the HLA alleles found in the group that developed inhibitor with those in the group that did not develop inhibitor to determine if any of these alleles were associated with either more or less likelihood of developing inhibitor. We also looked to see if these associations were related to the type of F8 gene mutation or with the racial ethnic group that the person designated. Before Dr. Ahmed talks about the statistical analysis of this information, I want to give you an idea of what this HLA data looks like. This is a small part of the table of data we developed from the DNA sequences. The column at the left side is the subject ID. We don't know the identity of anybody in the MLOF database, only their ID number. Each row in the table has the HLA alleles determined for that subject ID. As mentioned earlier, we each have 2 alleles for each HLA gene, 1 from each parent. We don't know here which comes from which parent, but as you see, there are 2 columns for gene A, 2 columns for gene B, and so on. There are several different D genes and also E, F, and G genes. We extracted information for a total of 13 HLA genes. What I'd like to point out here is that we can see how different each subject's HLA profile is. Looking just at the A column, you can see many different allele groups. That's the first two digits. So we have 2, we have 23, we have 68, we have 31, we have 32, and 74. And this large amount of variation also occurs in other HLA genes. So there's a tremendous amount of variation and diversity in the immunological makeup of each person. To analyze all this information, we arranged it into another table that has a separate column for each allele that is found for the entire group. So across the top here, instead of the gene names, we have one column for each individual HLA allele. The subject IDs down here, and the information in here represents whether that allele is present or absent for that subject. So now I'll turn it over to Dr. Ahmed to discuss the analysis performed using this information and what we found.
All right. Thank you, Dr. Contenti, and good afternoon, everyone. In step 2, we used several statistical analyses to analyze and describe the raw data. Data analysis were conducted using SAS, Statistical Analysis System, version 9.4. We evaluated the entire severe hemophilia A cohort, representing 1,841. We conducted also subgroup analysis based on factor VIII gene mutation, race, and ethnicity. We evaluated 540 HLA alleles that were obtained from the whole genome sequence data. So for the 540 alleles, we conducted pairwise analysis to assess the association between each presence of allele and inhibitor development. The association was tested using chi-square or Fisher-Exact test when appropriate. Association with p-value less than or equal 0.05 were considered significant and evaluated further by our regression analysis. Next slide, please. So for our regression analysis, we used binary logistic regression models to explore how each identified or significant allele at a significant level of 0.05 relates to inhibitor development in the entire severe hemophilia A cohort and in each subgroup. The regression models included each significant allele and the following variables: mutation type, race and ethnicity, age groups, factor VIII replacement therapy regimen, US geographic region. Next slide, please. So the next slide is shows our pairwise analysis finding for the entire severe hemophilia A cohort. So out of 540 alleles, we identified 20 alleles were associated with inhibitor development. 10 were associated positively with inhibitor development, and 10 inversely associated with inhibitor development. So to the left of this diagram, you'll see severe hemophilia A individuals with any of these 10 alleles were more likely to develop inhibitor. And to the right of this diagram, you'll see severe hemophilia A individuals with any of these 10 alleles were less likely to develop inhibitor. Next slide, please. So, and here this slide present our regression analysis finding for the entire severe hemophilia A cohort. So out of 20 significant alleles that were assessed in the regression after controlling for confounding effect, we identified 11 alleles were associated with inhibitor development. 4 positively associated, 7 inversely associated with inhibitor development. To the left of this diagram, you will see that severe hemophilia A individuals with any of these 4 alleles were more likely to develop inhibitor. To the right of the diagram, you will see severe hemophilia A individuals with any of these 7 alleles were less likely to develop inhibitor. With that, I will turn it back to Dr. Contente.
Thank you, Anwar. After our initial findings for the entire cohort, We next regrouped the data to exclude individuals who had a missense mutation. Although the factor VIII protein expressed doesn't function for this group, it means that the factor VIII treatment they receive is not usually perceived as a foreign protein, so inhibitor is less likely to develop. The incidence of inhibitor in the missense group was about 12%. The incidence of inhibitor in the not missense group was higher than in the entire cohort, about 34%. And we found a larger number of significant alleles than we found for the entire cohort. There were 15 in total, with 8 having increased risk and 7 having decreased risk. We also wondered whether having 2 alleles in this group would have any effect on the incidence inhibitor. What I'm going to show you next is the inhibitor incidence data for these 15 alleles in the form of a heat map. A heat map uses a gradient of color to represent intensity, with blue being cold or lower incidence of inhibitor, and red being hot or higher incidence of inhibitor. It will look a bit daunting at first, but I promise it will eventually make sense. Here we go. The 15 significant alleles that we identified are listed along the right side here. And in parentheses next to each allele is the number of times that it was found in the not missense group. The alleles are listed in increasing order of the inhibitor incidence associated with them. The first 8 have incidence lower than the group average, and the next 7 have incidence higher than the group average. They are also listed in that same order from left to right across the bottom. You can see that it is a lower inhibitor risk, bluer at the upper left, and a higher risk, more red, at the bottom right. The numbers along the diagonal, where the dotted line is, represent the inhibitor incidence found for that allele itself. For example, the incidence of allele DRB1 140101G, we move along here to the diagonal, is 18.75%. For the allele DQB 060201, which is down here, we move across and we see that the incidence of inhibitor is 41.42%. Please note that the number along the diagonal does not imply that the allele occurs 2 times in the same person. It's the incidence based on the number of times that allele occurs in the population. The boxes that are not on the diagonal do represent the percent incidence of inhibitor where 2 alleles are present. There are many gray boxes with NA. That means either we did not see any individuals who carried both of the alleles, or there was only 1 person. There are some notable changes in the inhibitor incidence associated with combinations of 2 alleles. We're going to look at 3 examples. We look at DRB1 1513 and go across, and we see that the incidence is 20.37. DRB1 040101 has an incidence of 23.81. And if we look at where the 2 meet, we go down from 1513 and across from 1401, we see 7.14. There are 14 people in this group who have both of these alleles, and the incidence of inhibitor percent is lower than either allele alone. If we now take another example and look at FO104, which is 50% incidence, and DRB1 150101, which is 39.59, we go down from here until we meet FO104, we see that the incidence in this group is 75%, and there are 8 people in this group. For the last example, using 1 increased risk allele and 1 decreased risk allele, we look at DQB1 060201 has an inhibitor incidence of 41.42%. We go up to look at DQB050301, which is 19.74%. We move down until we meet the line with DQB060201, and it's 11.11%. It appears that the increased risk effect of one allele could be reduced by the presence of the decreased risk allele. All right, what about all the other subgroups like F8 mutation, race, ethnicity? We carried out similar statistical analyses for these and made heat maps, which I won't show. But what I will show you now is a summary of all the subgroups we examined, presenting all the alleles that we found as significant, and using a statistic called odds ratio. This table summarizes all the significant alleles we found and how they distribute into the subgroups we examined. The number presented in the table is the odds ratio. This is the odds of inhibitor developing when that allele is present. An odds ratio of 2 means inhibitor is twice as likely to occur, and 3 means 3 times. When the odds ratio is less than 1, it means it's less likely to occur. So an odds ratio of 0.2 means inhibitor is 5 times less likely to occur, and an odds ratio of 0.5 means it's 2 times less likely to occur. The left-hand column is all of the alleles we found that were significant. Not all alleles were significant in all populations, but there are overlaps. Like the heat map, blue indicates lower risk with odds ratio less than 1, and pink is higher risk with odds ratio greater than 1. We already presented the entire cohort results and the not missense results here in column 2 and column 3. Again, the number that you see next to the odds ratio is the number of times the allele occurred in that population, and the number of subjects in each population is also presented. For the subgroup with intron 22 inversion, there were several alleles seen that were overlapped with those seen in the first 2 groups, but there were also 12 additional significant alleles found. For the group that has the missense mutation, we found 2 alleles, one lower risk and one higher risk, and these alleles were unique to that group. For the race ethnic subgroups, there were 3 alleles identified in the Black non-Hispanic category. One of them overlapped with the intron 22 category. The other 2 were unique. They did not overlap with any other group. In the white non-Hispanic group, there are 10 significant alleles. Most of them overlap with another group, either all severe, not missense, or intron 22, but there is one that is unique. I'll now turn it over to Dr. Pratt for the summary.
So in summary, that's a lot of data summarized there. We saw some distinct alleles associated with inhibitor risk for the various subgroups. In the white non-Hispanic cohort, there were higher and lower risk. In the Black non-Hispanic cohort, a smaller number of alleles, which does not reflect lower risk. It just reflects the fact that the cohort of Black patients was much smaller than that of white. We did not find any associations of HLA with inhibitor risk for the Hispanic white cohort. Again, this is probably because, almost certainly, because that cohort was smaller and quite genetically diverse. Interestingly, as Dr. Contente just described, there were specific combinations of higher and lower risk alleles that significantly affected the risks associated with the individual genes. So in conclusion, the class 1 genes are associated with innate immunity, which is our built-in defenses. The class 2 are associated with adaptive immunity, which is our antibody response to invaders. We found that both of these types of HLA genes are involved in reactions to factor VIII treatment. An unexpected result was that fully half of the alleles that we found that associated with inhibitor risk, either positively or negative, were class 1 alleles, and that means they mediate innate inflammatory type of immune responses, not antibody responses. This finding is also consistent with some recent studies, including a couple from my lab, that showed other innate immune messenger molecules in blood of individuals that have a current inhibitor. Our overall study confirmed some observations made in earlier studies, specifically associations between HLA DRB1 1501, 0602, and DQA 0102 with inhibitor risk. But in our study, these associations were significant only in the white cohort. Some of the earlier studies were carried out in England or in Europe with also a predominantly white cohort. Future studies with larger cohorts are needed to uncover additional relevant genetic risk factors in various populations. So take homes. The Black African American and white severe hemophilia A individuals in the US showed different HLA alleles associated with inhibitor risk. We saw evidence specific alleles were associated with increased or decreased risk. Innate immune mechanisms seem to be involved. For future directions, we still need to learn so much more about the unusual immunogenicity of factor VIII, which is more immunogenic than many other therapeutic proteins. I think the methods used here are applicable to studying other anti-drug antibody responses. So I think people outside the hemophilia community may have an interest in carrying out similar studies, especially looking at the risk associated with combined alleles. There's relevance of these studies to new therapeutic factor VIII products, including those delivered via gene therapy. There's still a compelling need for new approaches to promote peripheral tolerance to protein drugs. Peripheral just means immune. So what at the end of the day, what could this mean for the hemophilia A community? Well, this work is a small first step. Collection and analysis of information from additional larger populations within the US, outside the US, would strengthen the data. Associations with HLA with inhibitor risk also represent a first step toward a longstanding goal of developing polygenic risk scores that would include HLA genes and other genes that could allow healthcare providers to make more informed assessments of the risk of inhibitor development. I want to say very clearly that inhibitor development depends on environmental risks as well as genetics. Genetics will never explain everything, but it's important. An understanding of immunodominant HLA alleles and their cognate epitopes. Remember, those are the parts of the peptide, parts of factor VIII that bind the HLA. Could also be used in novel approaches to induce immune tolerance to factor VIII. And that could be the subject of another whole webinar. No time for it here. You have to just take my word for it that peptide therapeutics to induce tolerance are a very interesting branch of immunology. Some of the HLA alleles we found appeared to protect against or to either protect against inhibitors or correlate with lack of inhibitors, even when they are in the presence of higher risk alleles. It would be fascinating to better understand the mechanisms behind this that could then lead to novel approaches to develop mitigating strategies. So there's a number of people to thank on a study of this size. Our institution, of course, Uniform Services University, Andrew Frank worked with Dr. Contente on some of the bioinformatics. Drs. Johnson and Conkle had leadership roles in the My Life, Our Future biorepository. We're grateful to them and the entire My Life, Our Future program. Athen and National Bleeding Disorders Foundation were essential. Shelley Fletcher at Bloodworks Northwest sequenced the factor VIII genes for My Life, Our Future. Biogen, now BioVeritiv, now Sanofi, provided funding. Funding was also from the NIH, and the genomes themselves were from the National Heart, Lung, and Blood Foundation TopMed Consortium. And finally, as always, we very gratefully acknowledge all the blood donors and clinical staff whose contributions made these studies possible. So I want to put in a brief plug here at the end. So we have submitted an abstract on this work to the American Society of Hematology. Hopefully it will be accepted. And so if some of you are attending ASH, you could hear more there. We have a manuscript describing this work that has been accepted in the Journal of Thrombosis and Hemostasis. It'll come out after ASH due to the embargo. And finally, there's a prospective study launching now that is the inhibit module of the American Thrombosis Hemostasis Network Athens Transcends program. That's a big mouthful. There's a sub-study called Inhibit that will prospectively enroll young children and obtain blood samples and data starting with their first factor VIII infusion. This has not been done in a large way before. I encourage all of you to participate or talk to others about this so that we can get enough participants to do statistics on like we showed you here. So we'd be happy to take questions. Thank you.
Thank you very much, Dr. Ahmed, Dr. Contente, and Dr. Pratt. It's really interesting to hear about your investigations of these different genetic factors and how they might tell us something about why some people develop inhibitors and others do not. And Dr. Williams, thank you so much for putting all of this research into perspective for us. We really appreciate that. We'll go ahead now with our live Q&A, and we have a couple of different questions that have come in. Maybe we'll start with a couple of sort of technical questions around your study. Somebody asked if you could explain, you talked about looking at missense mutations or not missense mutations in the factor VIII gene, I believe. And could you just clarify what does that mean and why is that relevant?
Sarah, do you want to take that one?
Sure. The clinical data provides what the F8 mutation is, and there are a number of people who have missense mutations, meaning they have a change in the amino acid makeup of their F8, which makes it not function properly. But as we said, those people can sometimes do and sometimes don't have the immunological reaction to factor VIII. They're in that group, about 12% of them do. In the entire group, it's about 30%. So what we wanted to do was look at everybody in the group except those missense mutations. So that's why the group is called not missense.
Right, okay.
It includes all the mutations.
Okay, and so missense is when there's a change in the sequence of the protein. And so these are those that do not have, it's not a change in the sequence of the protein when you say not missense. What about a mutation that occurs in the promoter region or that causes like premature truncation? That can also affect the function of the protein. Were you able to query those types of mutations and whether they have an impact on the chances of somebody developing an inhibitor?
I'll take that one. So we went into that a little bit in our previous study that Dr. Ahmed and I published in, I guess, a year or two ago. And we found Dr. Ahmed's statistical analysis found, interestingly, that a number of mutations that were large changes in the factor VIII, for example, a truncation, stop codon, some of these very large ones, they all had a similar inhibitor risk. So we basically lumped them together in this study.
Okay.
I want to say that another very nicely done study by Samantha Gao, a large meta-analysis, found that intron 22 inversions had a lower inhibitor risk, somewhat slightly lower inhibitor risk compared to other large changes in the gene. And the My Life, Our Future repository had a different but also larger number of subjects, and we saw they all had similar inhibitor risks. So for the purpose of this study, we lumped them all together.
Okay, makes sense. Thank you. Another sort of basic technical question. You talked about the whole genome sequence. What does that mean and why was it important to use that data?
Sarah, go ahead.
Okay. It means that all of the sequence was gathered. A lot of times when doing diagnosis, only the regions of interest are sequenced. This way, we were able to get the entire part of chromosome 6 where we know all the HLA genes exist. So that we're looking at all the information that's available, so we can get all the HLA alleles out.
Okay, so now my PhD supervisor would hate me if I didn't ask, did you look at introns as well?
No, the HLA program is to recognize the different alleles so that you can classify them into what they are. So this just reports what allele is present. Now, we did not specifically look at sequence. We looked at what allele is present and what effect might it have.
So maybe I can ask another basic question then, like what's the difference between an allele and a gene?
Yeah, we use those interchangeably. For the purpose of this. I do want to say that we're getting some interesting questions here. There's a lot more data in the whole genome sequences that we did not access for this study, but that certainly could be accessed. So for example, some people have hemophilia A, severe hemophilia, and there's no mutation found in the coding region.
Yeah.
And so if you look more closely, you can find find something elsewhere in the genome. But there are a number of other projects that could be carried out using whole genome sequences.
Yeah.
So Fiona, before we go on to some of the other questions that are being put in by the attendees, I'd really like to get Mosi's thoughts about what he just heard and how he thinks this could potentially, or what kind of impact this has on him.
Thank you. Thank you all for the talk. We really appreciate it. And I'm trying to kind of break down these concepts as a layperson. I had a question. One question was in terms of when we think about ethnicity, how in the study was Black or Hispanic defined? Was this just for the person to decide demographically, or how did you see it, like in terms of does Black mean African American, or if there were other individuals in the United States, Jamaican, or in terms of Hispanic, is that Latin descent, Mexican? How was that defined in terms of the study?
That's a Sarah question.
Okay, we based it on whatever was in the data that we got. And so it was self-identified. So it said Black or African American. And then it said Hispanic or non-Hispanic, basically. Those were the choices. Of course, so there are many other races in there, but those groups were not large enough for us to do any meaningful statistics on. So we just focused on the groups where we had enough data. We took the groups as they were self-identified by the person in the study, and then we did a principal component analysis on that data and looked at the variation. And the standard is if you vary more than +3 or -3 from the mean of that number that's calculated, it means you're kind of an outlier. And we only found just a couple that did not meet that. So our groups are basically self-identified.
Yes, and I wanted to also add here, My Life, Our Future is a large and diverse database, but it is genetic data is not fully represent all the ethnic groups. So due to the small number for the other like ethnic groups, it may affect the power of the calculation. So that's why for like some genetic variant, they may not be significant, but actually we need the larger groups so we can assess the association.
Thank you. Okay.
Go ahead, Mosi.
Go ahead.
So if I could follow up on this a little bit, because this is so fascinating. Race and ethnicity, as we know, is a social construct, and we're overlaying on this a more medical construct to race and ethnicity. So I think it's great that it was self-identified, that people are saying, yes, I am Black. It's not being assigned by somebody else. But how do we include this social construct when we're looking at genetic information?
I agree that race is a social construct, but populations of people who were many years ago developing in one area developed different changes in their DNA from groups in other areas. And I
Okay.
think that those are just differences. They
Okay.
don't define race. They just define that different populations develop differently. And now with globalization and people mingling, that it's very hard to say, you know, Black, white, Asian. I mean, you can have so many different backgrounds. So it's going to be harder and harder to tease these out, trying to do it based on race or ethnicity.
Yeah, thanks, Sarah. That's a great explanation. I really appreciate that clarification.
Yeah, so we often hear that we're 99% of our genome, more than 99% is the same, right?
Right.
But the 1% that's different is mostly the immune response genes. And different
Fascinating.
populations that developed in different parts of the world had different immunological challenges, and that influences how their HLA responded. So it's more geography than race, actually, in some ways.
Absolutely. I think, and my next question, I guess, is in terms of like distilling this down for the community members such as myself. And I'm learning a lot from this in terms of the comment you made, Dr. Pratt, that blood without factor VIII is 10,000 times less efficacious than blood with factor VIII in it. I mean, just to hear that and to learn that. And so one of my questions is for the community, and like in distilling this down for us to understand, and parts of it genetically aren't things that we can do. And some of the inhibitor development is environmental. And so like, what's the next step for somebody to do? Like, can I avoid, and is there anything I can do to avoid inhibitor coming back? Because that's something that we don't know about the development, and we're sometimes paranoid about changing products and so on and so forth.
I think that's a great question, Mosi. Before Kate addresses that, as a clinician, my question, of course, is for all my new babies, should I be HLA typing them?
So the short
Say
answer,
yes.
the short answer to that is it used to be quite expensive and labor intensive to
Right.
genotype. Now you can get a whole genome sequence for about $200, and it's going to get even cheaper. And it's straight. Am I allowed to say it's straightforward to pull the HLA out, Sarah?
Yeah, it's doable.
Straightforward with the proper expertise.
With the proper expertise, yeah. And you didn't jump in at all. I was about to say for this question, Dr. Reck should respond. But as far as, mostly there have been studies in the past of people wondering, worried about vaccinations at the same time as getting, well, it turns out that both in animal model studies and epidemiological studies, there wasn't any difference in inhibitor development close to or farther away from vaccines. People have wondered whether breastfeeding versus formula would make a difference. No difference there. So it's a challenging problem. That's why we've been working on this for a while.
And I think Mosi's point of view is so interesting, particularly for me as a pediatrician. Like, when someone is tolerized after treatment, I'm like, okay, go to your adult provider. But I don't think about recurrence a whole lot at all, inhibitor recurrence. Do you think the data that you presented today informs recurrence at all?
We did not look at the age, we didn't do a sub-analysis of the age at which it developed, and also this was the inhibitor data was historic. It was from
Yeah.
records.
Right.
This is why something like, not to plug too much my own study, it's me and a lot of other people, the inhibit module
Inhibit study
will
is
be
so
looking
important.
at the immune response during the initial infusions. And there have been some small studies there, and ours will be fairly small, but a little bit bigger. And I think we'll get some very useful information there, because we've always been looking backwards to inhibitors that developed before we got the blood samples to study.
Well, I was lucky enough to be an investigator on the HIPS study, which has been, has had some really fascinating results come of it. But again, we only had 24 human beings on that study. So we're hoping that inhibit is going to be enroll more people.
And there's also the HARP study coming up that's going to be enrolling mother-child pairs with Jill Johnson as PI. So there are there's several more opportunities to participate in research.
One question that did come also from the audience, Amy Dunn has asked if you wanted to say anything about also the high doses of factor encountered in a surgical situation, whether you want to say anything about that and how that pertains to risk of factor of inhibitor development or maybe recurrence of inhibitor.
I think that might be a Dr. Reck question again.
So that's interesting, right? So that goes back to the environmental risk factors for inhibitor development. I think this plays in exactly to what Kate was talking about, this. You said polygenic risk score. I would say poly-life risk score, right? Not just the genetics that put people at risk, but all the environmental risks that we know about. Going all the way back to Donna D. McKelly's North American Registry.
And it's frustrating that take somebody that's on emicizumab or something similar, and they're getting along just fine without factor VIII. Now they fall off their bicycle or get in a car crash or something, and they need factor VIII right in the inflammatory environment that you don't want.
Right.
So they need it at a time where they're at high risk, and there's just no getting around that. So it would be better if they had been tolerized before the car accident or the surgery or whatever happened. And I think there is still a case to be made for trying to tolerize, especially young kids, to factor VIII, whether it's via infusions. I sure wish that oral tolerance would come online. I guess there is not. I don't know the details. There's been no funding available apparently, or no industry. No industry has taken up the promising results from Roland Herzog and collaborators, where they express factor VIII in lettuce, and it could be turned into pills that you take to attempt to tolerize. Who knows if that would work, but that would be wonderful if there was some straightforward way to tolerize young kids so that then later when they were exposed to factor VIII, it would be less risky.
Yes, and that's exactly what Amy Dunn is saying as well. This is really an important point for the pediatric community of people who are on emicizumab, for example. So maybe we need a whole nother webinar on that. Maybe we can throw that out to the universe.
I think Amy just volunteered for that webinar.
Oh, there we go. All right. Well, we'll be booking you, Amy. Unfortunately, that gets us to the end of our hour. I feel like there's so much more that we could discuss here. But I really want to thank all of our guests, Drs. William, Pratt, Ahmed, Contante, and Reck, for sharing your time and your expertise with us today. It's been really great to hear from all of you on this topic that's really very important to a lot of people. And thank you so much also to all of our audience for being here today. We appreciate you. This recorded webinar will be available shortly on bleeding.org under the events tab. If you select past events, you can find all our archived webinars there, and also you can see upcoming webinars. We're going to take a little break now for the next couple of weeks as everybody is busy with the NBDF Bleeding Disorders Conference, August 21 to 23, and then just enjoying the end of summer. So please do come back and join us in a couple of weeks' time, September 10th, and we will have a hemobiologics sponsored webinar featuring Dr. Guy Young. So thank you so much for joining us today. Have a great couple of weeks and enjoy BDC.